Southampton for the DNP meeting, and yeah.
Right. Okay, we're being recorded now, so.
Yeah. And also out at the, well, what did you have? You had another meeting there out at that zoo.
We had, that's right, yeah, yeah.
Mm-hmm.
That was, well, what's that actually? I think
Of some sort, yeah.
Yeah, it was one of the meetings of, well, we've had meetings of this Zero Field Network there. I don't think you were at that one. I think, oh, we had like what's called a doctoral training school, like PhD programs. And there was like a final meeting of that. I think maybe you came to that one. If I remember.
Mm-hmm.
Yeah, I think we had you in for that one. Yeah.
I remember Shimon was there, yeah, as well.
Yeah.
But, yeah.
That's right. Yeah. Yeah. Yeah, it seems like another world now.
Mm-hmm.
You know, with how is it over there in the States now? Are you free to do what you want now, or?
Well, in Massachusetts, the vaccination rates, what's it, approaching 70%, Klaus? Is that right?
Yeah. And in our town, it's 95%. So
yeah.
We're really approaching herd immunity, and all the restrictions are being lifted.
Yeah.
And the numbers look very, very good.
So yesterday in the state of Massachusetts, there were 44 new cases of COVID. So it's, you know, in January, there were 8,000 one day, you know, so it's dropped. The vaccination program has been very successful. Yeah.
Good.
So, and then at MIT, we were being tested, swab tested twice, 3 times a week. That's gone down to 1 time a week. And they've actually opened the gym again yesterday.
Wow.
So it's relaxing, you know. And I still wear a mask just out of habit, but many people don't, you know. And within MIT, if you're in a building, you're supposed to wear a mask still, but outside, you don't have to. So, yeah. So it's gotten a lot better. Yeah. So how's the vaccination rate in France and England these days, in the UK? Mm-hmm.
Well, the UK, it's pretty high now. It's, I think it's about 60% over the whole population.
Mm-hmm.
In France, it's about 45 or so %. But one of the statistics that keeps me busy is the number of people in intensive care units. And at the very peak, it was about 7,000 in the country, and it's still about 3,000. And the likelihood to survive once you're in intensive care is not too good. So I consider the situation is not yet resolved.
Yeah, there's this new variant going around, this Delta variant or India variant, which at least in the UK is causing a lot of worry.
Yeah, supposedly the Pfizer and Moderna vaccines are still effective against it. I read last night in the New York Times. There was an article in the New York Times that said that last night. So,
okay.
Yeah. So if you've had one of those vaccines, you should be still in good shape. So, yeah.
Yeah, I think maybe the AstraZeneca is a bit less effective. That's what I've heard, but I don't know the figures.
Yeah. So we start about 11:05 MIT time, 5 after the hour. Yeah.
Okay.
So
there are only 7 participants so far.
Are we in the main session now, or? Oh no, we have 66.
159, excuse me, attendees.
Yeah.
So Galia, how are things in California with respect to COVID? Have they calmed down any? I mean,
Yeah, the numbers are pretty good. I think California officially opens today, so there are
okay.
supposed to be no longer any limits on how many people can be at an event, and I think no masks for vaccinated people.
Uh-huh.
So yeah, I don't know what UCSD will decide. So we're waiting for new guidance on that, but yeah, the hope is that in the fall it will be everything in person classes, everybody will be back. So
Mhm. Yeah. Mhm. Well, that's certainly the plan here. Yeah. Mhm. Yeah. I mean, MIT is going to require people to be vaccinated to come to campus. Yeah.
yeah, I think we got yesterday an email that all of the UCs will be requiring students, employees to be vaccinated. So
Yeah.
yeah.
Yeah. So
That's interesting. That's sort of politically off limits in the UK, actually. That
yeah,
type of,
well, it was controversial here. I mean, there was a faculty meeting and some people were quite upset about it. I mean, they're religious, if they're medical or religious reasons, you can not take the vaccine, not be vaccinated,
okay,
but you have to present those. Yeah.
I see.
Yeah. Mhm. Okay, Kong, it's 11:05. Let's get going with your presentation, or you want me to just?
No, you can do it. I mean, like, yeah, there's nothing much to say except I mean, it's the same thing that I would just ask everyone to sign up to our mailing list, and I will just put everything in the chat room. So for whoever is interested in following our, in watching our past recorded sessions and signing up for mailing list, I mean, all the information is on the Griffin Group website. So feel free to check it out. Yeah.
Okay, so let me just share a screen today. So today, as many of you know, we had an unfortunate event in magnetic resonance in the community, and that is that Richard Ernst, who won the Nobel Prize in 1991, passed away last Friday on June 4th. He was born in Winterthur in August 1933, and as I said, passed away in 2021. So he was actually almost 87 years old. And he won, as I said, the Nobel Prize in Chemistry in 91. And for those of you who don't know Richard, or who hadn't had contact with him, and many people perhaps in this audience have not, he had a number of monumental papers that were really important, influenced the field, the directions of research that everybody goes through these days. Probably the first one was his introduction of Fourier transform NMR in 1966 with Ernst. And the paper was by Ernst and Anderson and published in Review of Scientific Instruments. It was interesting, the story he told about this, and that they sent it to the Journal of Chemical Physics first, and the paper was rejected because it was too elementary, supposedly. So they sent it to RSI, and of course, it's become the foundation of many different areas of spectroscopy as well. And then in 1976, he published this paper by Auer, Bertholdi, and Ernst on two-dimensional NMR, which was the beginning of multidimensional spectroscopy, not only in magnetic resonance, but in the optical regime and in many other areas as well. And in 79, Beat Meyer, when Beat was a graduate student, he was introduced to an idea by John Jenner. You do not just two pulses in an experiment, but you do three pulses, and you have a mixing period in between. And that's the so-called EXI. It was called an EXI experiment, an experiment to examine chemical exchange processes by two-dimensional NMR, and we know it today as a NOSI experiment. And in fact, this particular experiment sort of nucleated another Nobel Prize, the one won by Kurt Wittrich, subsequently on protein structure. Now, I first met Richard, in probably 1971, I think, at a Gordon Research Conference up in New Hampshire. But I used to see him at many, many meetings. And the earliest photograph that I could find of him was this particular photograph right here that I took of he and Alex Pines. It was at a Congress Ampere. That was before Euromar was established in Tallinn, Estonia in 1978. And of course, I've seen him, saw him many, many times subsequent to that. You know, there was, as he liked to say, T1 relaxation occurring. So this was the same photograph, which he insisted be in front of this trash dumpster in Berkeley, California, in about 2000 or so. And then we were together at John Jenner's 60th birthday and his 70th birthday party in Zurich subsequently. Now, a few years ago, I was fortunate enough to receive the Richard Ernst Award at Euromar. And at the time, I wrote Richard and asked him if there was something I should show in his honor, something that he would appreciate very, very much. And so he sent me this poster, which I learned this morning from Kong, that is still in the ETH near the NMR room, the ETH. And it says, science can be captivating. So we really want to thank our good friend and mentor, Richard, for his inspiring research and his very entertaining leadership in magnetic resonance. And as Rob Tico was quoted yesterday in Chemical Engineering News, once Richard won the Nobel Prize, Richard was a really nice guy. He was a very caring person. He was a great scientist, a good mentor, and a good friend. And once he won the Nobel Prize, he tried to use that attention to express his ideas about how to make the world really a better place. And as I said, this appeared in an obituary that was in CNE News yesterday, that arrived in my inbox yesterday. So now we're going to have two more comments about Richard. The first from Malcolm Levitt, and the other from Jeffrey Bodenhausen, who were both members of Richard's research group in Zurich. So Malcolm.
Yeah, thank you. I won't take up much time. I really don't quite know what to say. Richard's, I had the good fortune of being a member of Richard's research group in Zurich between about 1982 and 1985. I think I finally left that group because some strange American guy came up to me and said if I wanted to come to work at MIT. And that strange American guy was Bob Griffin, actually. And the rest is history, as they say. But working with Richard was, something you don't forget. Being able to absorb the atmosphere of a really truly first-class scientific environment with very, very deep theory, which was way over my head, most of it at the time. I really felt very small, I think, when I first joined that group, and just a little bit larger by the time I left. But one picked up the feeling of how to do really serious science, but in a very creative and fun way as well. I have a couple of anecdotes which I think maybe express. There are many stories to tell, of course, and I suspect that Jeffrey may tell a few more of them. But there were a couple of things which made some particular impressions on me. Actually, one thing, I was just reflecting on this, actually. I do remember when I heard that Richard won the Nobel Prize. It turned out I was in Stockholm at that time, just across the road, in fact, from where the Nobel Prize is awarded. And then I read the citation and realized it was being awarded for Fourier transform NMR. Well, at that time, I'd been thoroughly immersed in Richard's work, of course, all the 2D NMR, the enormous developments, the invention of FT imaging, solid states, multidimensional spectroscopy, populations, coherences, density matrix, it goes on and on. It's part of the fabric of the way we all think and what we know about NMR. And my instant reaction when I heard that the Nobel Prize was for Fourier transform NMR, my first reaction was, well, I don't know, that's sort of trivial, isn't it? You know, get a Nobel Prize for that, just doing single pulse or something. Of course, reflecting on it now with the advantage of maybe a bit more time and perspective, what I was reacting to at that time was probably a common problem with this type of great science, is that it becomes very soon the absolute complete fabric, the water in which you swim. So you don't even notice it anymore. So the idea of doing a pulse and then taking Fourier transform to get this spectrum, just even at that time in the 1990s, then we're talking about, was just what I was brought up with. So it seemed very almost trivial by then, just because it had become completely the natural way of doing things. And of course, it needs that perspective. Now we can look back and think, well, wait, in 1960s, NMR was basically crawling through a field sweep with a pen moving across a piece of paper to indicate where the peaks are. It was a completely revolutionary thought to do a single pulse and then use computer power to process the data and generate an NMR spectrum. And there are many anecdotes about how long that took in the first few attempts in Palo Alto, with paper strips being driven or walked across the road to the only computer which was capable of performing a Fourier transform, which then took a whole week or something like that. So it needed tremendous vision and insight to initiate this revolution. And then once we're now living, the rest of our careers have been living through that revolution, which Ernst initiated, and maybe we can hardly see it anymore. I have a few more anecdotes, but there's one which I'd like to say, which is, I think, revealing, because it shows that despite Ernst's greatness as a scientist and a visionary, he had some weaknesses as well. And knowing him, I knew him well enough to realize that he would appreciate his weaknesses sometimes being pointed out. He was quite a humble person who knew his own limitations. This is, I think it's an interesting story. When I was in Zurich, I started to be interested in trying solid state NMR, which I'd never done before. And at the time, actually, that was regarded as being a very fearsome and very, very difficult field. But I thought, okay, I'll try a few things. And I soon realized that it would be really useful if there was something like isotopically labeled enriched material, which I'd heard, I think, somehow that some, guys with huge resources in America or something had actually got hold of some carbon-13 enriched material, which was almost unheard of at the time. And I thought this would be really cool, because it would save so much time in tuning things up and so on. And I asked Richard, can we purchase some of this carbon-13 labeled stuff? And he said, no, absolutely not. And in fact, during all the time I was there, and I'm not sure if Jeffrey can back me up on this, the use of isotopically enriched material was completely forbidden. And why? Well, the reason is very revealing, actually, for Richard. He wasn't just somebody who wanted to play with fancy concepts or abstract theories. He wanted to be useful in the world and to the community of scientists. And his view at that time, which turned out to be profoundly wrong, I'm sure he would now be the first to admit it, was that very few people could afford to get hold of enriched material, which was very expensive at that time. And therefore, we should not fall into the temptation of using such material, because then we would end up developing techniques which nobody would use. And of course, it's a very rare example, actually, of him being completely wrong and his vision failing him. Of course, he couldn't foresee or it was hard to foresee that the price of these things would come radically down. And indeed, now we could hardly imagine most forms of NMR, I think, without using isotopically enriched material. But it also gives, I hope, a flavor of him, that he really wanted His ambition really was to do great science, but also to be profoundly useful. And that colored everything he did, actually, within science and also beyond it. So it's a shame for those of you who never met him and never saw him speak. But I hope that gives you a little flavor of what sort of person he was.
Okay, thank you very much, Malcolm. Jeffrey?
Yes, that was very nice, Malcolm. Actually, I knew Richard a bit earlier because I was an undergraduate at ETH between 1970 and 74. And in this last year, 74, I actually did sort of a degree, what is it called, a master's. It used to be called a Diplomarbeit in German, in his group. And in fact, at the same time as Alexander Wokan. And I remember sometime in April 74, Richard gave a lecture just to the two of us about relaxation theory and red field theory, and I was mightily intimidated. But then I didn't like it very much because at the end of 74, I decided to emigrate to Oxford and to do a PhD with Ray Freeman, basically because I thought it was less scary and less intimidating. And in fact, I had asked Richard's advice on this, and he confirmed that Ray would do rather simple-minded things, which is not completely wrong. Now, I also remember Richard when I came back in 1980, between 1980 and 85. I remember him as a very complex person. Of course, he was ambitious. He wanted to be successful in science. He didn't dislike being accepted socially as well. And after his Nobel, of course, he became something like a national hero. And he seemed to enjoy that. But his focus was mostly on good science rather than on society. But he had other interests, and one of them that was pointed out to me by Herbert Kogler, who listened to radio a lot. And there was a competition on the Swiss radio where they played a piece of classical music, I believe mostly Baroque music. And the listeners were invited to guess who the composer was, and preferably which symphony or whatever. And Richard won this competition week after week after week, until in fact the whole competition was just suspended because it was no fun for the other listeners. And I asked him about this many years later in ZOOMinar, and he said, yeah, sometimes it was a bit tricky to remember the difference between Haydn's 41st and 42nd symphony. He wasn't quite sure which theme belonged in which of these two theaters, but otherwise it was very easy. Now you should know that there were several hundred composers in the 18th century in Germany and Italy and France. So he must have a very good memory. And also I remember that in his library at home, he actually had a lot of sheet music, printed music, and he would read music rather than listen to it, which is also rather unusual. He also had an interest in Tibetan art. In fact, he had one of the largest private collections of Tibetan paintings in his home, and which he had partly bought with the money of the Nobel Prize. But Tibetan paintings was not just about color and about the skill of painting characters. It's also a sort of a mysterious representations of hundreds of Buddhas and Bodhisattvas or whatever they're called. And Richard was skilled enough to read these paintings. He could really understand what is going on in them and what is being represented and what mattered, and whether it was about life and death or about the universe or whatever. He had for him it was a transparent text, which is not given for everybody. Now, of course, I've also known Richard as the manager of his laboratory, and there I think he had very different attitudes. To some people, he could be very authoritarian, even rather nasty. And to others, he gave them long lists of things to do, like tune the proton preamplifier and then do the carbon amplifier, etc. Lists which are pinned on the wall. Of course, that was not my flavor. But I think it's roughly at the time of my arrival, or maybe Malcolm's arrival, that he changed his attitude completely and realized that it was more fun to listen to people rather than boss them around. And certainly the recollections that I have is that he listened a lot and he was quite willing to absorb contradictory statements. Now, he also had heavy responsibilities. Nobody knows about this, but at ETH, he spent many years as president of the so-called Research Commission, the Forschungskommission, which dishes out something like $1 billion a year to different research projects. And it was his responsibility to decide where this kind of money would go. And he never complained about that, but it's a major, major effort. Then he was famous for his lectures. In my recollection, they were always too long. He always prepared something like 50 or 100 transparencies in those days. And then he put the first 10 transparencies on the projector and commented them very ably, very interesting. And the remaining 90 transparencies remained on a table and we would never know what's in them. And that was a bit frustrating. Nevertheless, he was very successful. And I remember at Asilomar, that's in California, there are actually two separate halls. There's Merrill Hall, which holds about 1,000 people or so, and a smaller hall called the Chapel, which has about 400, 500 people seats. And it used to be so that they made a video connection between these two until they realized that there was only one point that this connection was really necessary, and that was during Richard's lecture. Richard, by the way, gave a lecture every year, along with Ray Freeman, and these two were the only two who were allowed to make an exception to the rule, and the rule of ENC was that there should be different speakers every year. But this rule, as I said, was not applied to Richard, and Richard came back every year and always gave spectacular lectures. But as I said, the video was only necessary for his lectures. Nobody else could fill these two rooms. And at one point, when I was on the executive committee of ENC, I said, well, let's stop inviting Richard so that we can save the money on the video connection. And I remember this was followed by deadly silence in the room. How could you say such nonsense, not inviting Richard? So they continued to invite him, rightly so, and they continued to pay for the video connection. Now, I also remember the beginnings of the book that we wrote together with Alexander Volkan, Principles of whatever. And he showed me a first sketch of this, which he had worked on together with Alexander before I came. And he showed this in, I think, October 1980. And it was basically a rewrite of his paper with Auer, Bartholdi, and Ernst. And I gave the sketch back to him the following day, saying that I thought it was totally unreadable. And he was a bit upset. And he didn't speak about the subject again for at least a whole year. And then after a year, when we had done a few papers together and done all sorts of, had a lot of fun, he came back to me and said, would you maybe continue working on this book again? And I did. I think it was about 1983, and it took about 2 years working on this. And of course, the essential frame of it is his. My job was mostly filling in the gaps. And I gave him, I worked on it all week long, from Monday morning till Friday evening. And on Friday, I came to his office and handed over ceremoniously chapter such and such. And on Monday morning, he came to my office and handed over his comments. And this was a lot of work because I had spent an entire week and he only had spent one weekend. So he worked nonstop, around the clock. And he did a very good job, very thorough, very careful, very demanding. So I think I have a very great respect for this man. Thank you.
Okay, thank you very, very much, Malcolm. And thank you very, very much, Jeffrey, for those really beautiful talks, insights into life with a guy who is really important for all of us and has made this field really what it is today, right? Okay, so with that, those two presentations, again, thank you. Let's go on to the two talks for today. And the first one is by Klaus Schmidt-Rohr from Brandeis. So Klaus was an undergraduate in physics and mines, and then he did his PhD there with Hans Wolfgang Spies. And there he wrote a book too on polymer NMR, which is really an excellent textbook. He was a postdoc subsequently with Alex Pines at Berkeley, and then an associate professor at UMass Amherst and Iowa State. And about 5 or 6 years ago, he moved to Brandeis University. And today he's going to tell us about the determination of domain sizes of 10 to 200 nanometers in organic solids with advanced analysis of proton inversion recovery. So Klaus, the floor is yours. Okay?
Thank you, Bob, for the introduction, and thanks to you and Kong for giving me the opportunity to speak here today. I want to discuss domain size determination in multi-component polymers and other complex organic solids on scales between 10 and a few hundred nanometers. This is based on proton spin diffusion occurring during proton inversion recovery. Set that there. The experiment from which we get our data is very simple. Proton inversion recovery with cross-polarization and carbon-13 detection has been around for decades. What is non-trivial is to extract domain size information from the observed relaxation behavior. And one challenge consists in the limited knowledge of the intrinsic relaxation times of the components. Let's look at this inversion recovery simulated in a two-component system. It occurs on a time scale of seconds. And so there is simultaneous relaxation and spin diffusion from the fast to the slow relaxing component. As a result, the fast one is slowed down, the slow one is accelerated, and the observed relaxation is different from the intrinsic relaxation in a way that reflects domain size information, and that's what we want to extract. But the challenge consists in limited knowledge of these intrinsic relaxation times. We've developed various approaches to still obtain domain size information under these constraints. One is that we can get a lower limit, a minimum domain size, just from the observed relaxation time ratio. So if the ratio is 2, let's say, we go up to the curve for the corresponding composition and read off the lower limit of the domain size. If we have some uncertain information about intrinsic relaxation times, we can put that in a diagram like this. Again, the crucial experimental input is the ratio of observed relaxation times. So that would be that value of 2 here on this contour line. And then from the overlap range of this information, we can read off a range of domain sizes. And at the very end, I want to briefly mention an offshoot of this spin diffusion after inversion recovery that allows us to obtain domain sizes even without any input of intrinsic relaxation time information. Another challenge here is the high dimensionality of the parameter space. There are 2 domain sizes and 2 intrinsic relaxation times that affect the observed behavior. And I want to focus on the concept of the average relaxation time, which allows us to reduce the dimensionality so that a general analysis like this can be done with a few 2-dimensional graphs. Here I want to show some data to convince you that this effect of spin diffusion really exists. These are data for 4 different commercial polystyrene PMMA block copolymers. And we see that for small domain sizes, spin diffusion indeed nearly equalizes the observed relaxation, while for larger domain sizes, we start to get closer and closer to the intrinsic relaxation times of pure polystyrene or PMMA. I believe that this experiment is underutilized. We are finding many useful applications of it, and mostly starting with the simple principle that 2 components that have a different proton T1, like these carbohydrates here and the aromatic lignin residues, that they cannot be finely mixed. And the question of what different means and how many nanometers finely mixed is, that's what our quantitative analysis can exactly quantify. Here we're just using it to start with, to identify what domains are present, what composition they have. And then we can use the ratio of these time constants and put them into our graphical analysis and get at least a lower limit for the domain size, which says the layer thicknesses here must be at least 10 nanometers. I need to point out that these are not quick shots. You can't get these spectra in 64 scans because both intensities are attenuated near these 0 crossings that we choose. So one spectrum is at the 0 crossing of the fast relaxing aromatic lignin component, And then 50 milliseconds later, we record the red spectrum where the cellulose peaks are nulled. So the intensity is not large. You need overnight measurements, but you get information comparable to a head core experiment where you would invest a similar amount of time. And we used this useful information from inversion recovery to analyze the transformations that happen during friction welding of wood, which is a pretty fascinating process where a machine presses two pieces of wood together and then rubs them quickly for a few seconds. And a solid joint, a weld forms. They are firmly connected. And you see that quite intriguingly, it seems that the wood actually in some sense melts during this process. We've analyzed the chemical changes in that weld layer, the dark layer that connects the two pieces, and find an increase in aromatic components that had previously interpreted as an increase in aromatic lignin. Inversion recovery shows that that's clearly not correct. The new aromatic components are distinct from the lignin. They're clearly associated with the carbohydrate domain, and that in hindsight actually makes perfect sense. Under these conditions, this heats up to 300 to 400 degrees C. Aromatization of carbohydrates occurs much more easily than aromatization of lignin, which is already mostly aromatic anyway. Here briefly, a head core spectrum with spin diffusion of a similar type of wood. And we cannot, it is not useful, we don't see component resolution. So this is an example why sometimes it is better to seek relaxation time rather than proton chemical shift contrast. Here an example of a pressure synthesized carbon material. We've been studying a lot of such carbon nanothreads. This one is putative nanoribbons, a particularly complex material. We analyzed it in terms of its composition with spectral editing. And then we performed inversion recovery with carbon detection, and that revealed that this had been a meaningless analysis. The spectrum actually consists of two components with proton relaxation times that are an order of magnitude different. So we need to use inversion recovery, the zero crossings, in order to have a meaningful identification of a specific component. Here, a curiosity, these are spectra of a mixture of carbon-13 enriched amino acids that we made years ago for pulse sequence development. And we recently discovered that they all show distinct zero crossings, that we can separate these different components through inversion recovery with carbon detection. And this shows that organic materials with pretty similar chemical composition, pretty similar proton chemical shifts, and limited resolution in head core NMR may give you excellent relaxation contrast. The message is similar here with 4 components of industrial materials that we've been working on with our sponsor. I cannot show you the actual materials due to secrecy issues, but what you see is that these components have quite distinctive intrinsic relaxation times and provide us with a contrast for quantifying domain size from the inversion recovery. I want to now move to the analysis. It is based on our numerical simulations of simultaneous relaxation and spin diffusion. This allows us to test the limitations of the traditional rule of thumb analysis here. We can study both bilayers and periodic layered structures. I want to briefly discuss the relative merits of inversion recovery and other methods for measuring proton spin lattice relaxation. And then discuss T1 average, really the inverse of the average relaxation rate, why it shows up, that it's the time scale of our experiment, that we can experimentally determine it, and how we use it in data reduction. And you'll see all the scaling in our plots, which makes it possible to do this on a 2-dimensional sheet of paper. And then I want to go into these analyses from observed and estimated intrinsic relaxation times, and then explain how we get valid and meaningful stringent limits, lower limits for domain sizes without having to know intrinsic relaxation times. If there's enough time, I want to show how we validate this on these commercial block copolymers, and show you how we use headcore here to determine the domain sizes that we can then put into our analysis, and to check that it's all done correctly. In this context, we've developed a simple single spectrum referencing that allows us to use headcore to measure domain sizes up to the wavelength of light if we have good enough signal to noise. Then I want to summarize, and at the very end briefly give an outlook to an experiment that is an offshoot here, spin diffusion after inversion recovery, where we can get domain sizes without knowing the intrinsic relaxation times. Most of you are familiar with proton spin diffusion down a magnetization gradient due to dipolar couplings between spatially close protons. It occurs with a diffusion coefficient that gives us a root mean square displacement of the magnetization within half a second on a scale of a little over 20 nanometers. This corresponds to a radius. And so we can probe domain diameters of up to 40 nanometers. And that's what you see in these historical data. The longest example here is a long period of 80 nanometers or 240 nanometer domain sizes roughly. And it does not reach equilibration. And I want to explain also in this talk how we can expand into this larger domain region here. We use numerical simulation of the proton spin diffusion, a discretized version of Fick's law of diffusion. And you transform it, you get this simple equation, discrete in space and time. And you can interpret it as a simple rate process where neighboring protons share magnetization. And this has been around for decades. We now add relaxation, so we can simulate the simultaneous relaxation and spin diffusion during inversion recovery, and the term just carries through and allows us to calculate these spatial magnetization profiles as a function of time. We then integrate the total magnetization within component A and in component B, and that gives us these plots. In our collaborations with an industrial partner, we're looking a lot at polymer capsules with a multi-layer, in the simplest case, bilayer structure. And that's something that we can also cover with these simulations, because what we actually simulate is just this bilayer. And through reflecting boundary conditions, that becomes equivalent to the periodic structure. And we only need to take into account that the corresponding size of a domain in a periodic structure is twice that in a bilayer. We can also use our simulations to test the assumptions, the claims in the traditional analysis of domain sizes from inversion recovery, where it is stated that if you observe different relaxation of your components, the domains must be larger than the root mean square displacement of the magnetization within the relaxation time. And here are some simulations that show that that's not quite true here. The other prediction is that if the observed relaxation is the same, you must have small domains smaller than that root mean square displacement. And here's an example from large domains that still give you a very similar relaxation. And that all has to do, of course, with the intrinsic relaxation times. And I also note that this does not tell you anything if the relaxation behavior is similar. It's not that different, but not the same. And our quantitative analysis actually does something very similar to this comparison, a larger than, a lower limit, but in a way that quantitatively takes into account how big that difference or ratio of the relaxation times actually is. I'm focusing on inversion recovery, but of course there's also saturation recovery and plus minus Z storage for measuring relaxation times. And with these simulations, we can confirm that even when there's spin diffusion, all of these methods give you equivalent curves as long as you start with uniform initial magnetization. And they're all directly related. In practice, they have different advantages. In saturation recovery, you know exactly that you start with vanishing magnetization with the ±Z filtering. You know that you will end exactly at 0. Inversion recovery has the advantage of superior dynamic range, meaning that if we have some fluctuations in our measured intensities, maybe because our cross-polarization is not so stable, that has less of an effect here because the magnetization in the critical range is small, while for these other experiments, if we blur the lines here, we may not be able to really show convincingly that we have a different relaxation. And of course, the zero crossings, which I focused on earlier in this spectral editing approach, is a special feature of the inversion recovery. T1 average is a central quantity in our analysis. It is the inverse of the average relaxation rate. It is well known in relaxation theory that you need to average the rates, not the relaxation times, and the fractions of the components are the weighting factors of these different rates. And we observe T1 average in experiment when we have very small domains, as you can see in the simulation for 5 nanometer domains, where spin diffusion instantaneously equalizes the magnetization. And as a result, the observed relaxation of the two components is the same, even though the intrinsic relaxation is distinct. T1 average also behaves as an average in the sense that it's always between the short and the long relaxation time, whether intrinsic or observed. In the convention that we're using, A is always the fast relaxing component. We can prove this relation in our diffusion context and see under what condition it really holds by spatially integrating our differential equation of diffusion and relaxation over a full period. For mathematical reasons, the spin diffusion term goes away, but we can also understand intuitively that spin diffusion will not lead to change of the total magnetization with time. So this is expected. Only relaxation comes into play here. And now under the special condition of uniform magnetization, we can further simplify this expression into a very simple relaxation equation for the total magnetization, where we see exactly the right-hand side here as the relaxation rate. Uniform magnetization is obtained when we have the small domains, as I had shown you with this almost instantaneous equalization by spin diffusion. And importantly, it is also the case in the initial regime in our inversion recovery. We apply an unselective inversion pulse. So initially the magnetization is uniformly inverted. And so the initial rate is T1 of the total magnetization, T1 average. That means we can determine it from our experiment by initial rate analysis. And if the observed relaxation is described well as an exponential, we can use this equation, the direct equivalent of the expression here for the intrinsic relaxation times, also with the observed relaxation times. And so we know T1 average. Here is an example of how it helps us in the reduction of our parameter space. These are two different simulations for two different situations with different relaxation times and domain sizes, but you see that they look exactly the same, that they're equivalent. And that's because we changed the relaxation times and the running time here by a factor of 9, and the domain sizes by a factor of square root of 9. And that leads to equivalent situations. We can then use reduced quantities, derive the time constants by T1 average and the domain sizes by square root of T1 average. Both situations give the same values and they collapse into one point in our diagram. So we're not wasting dimensions to repeat equivalent simulations. And that's what you see in our graphs, always domains divided by square root of T1 average and relaxation times divided by T1 average. This slide shows how in practice we get from that 4-dimensional diameter space parameter space to these two-dimensional plots with a third parameter that's effectively a third dimension. We have measured, and that's the values that we will later mark on these contours, the ratio of the observed relaxation times, and from quantitative NMR, we can get the fraction of component A. And then we systematically vary this ratio of the intrinsic relaxation times. For each value, we can calculate the needed intrinsic relaxation times at T1 average of 1 second. That's always our reference value. We vary also the domain size here, and through the, in this case, 50-50 ratio, we know that the other domain has the same thickness. So now we have all 4 parameters we need for numerically simulating the inversion recovery here. We record the 0 crossings and calculate their ratio and make that a function of these 2 variables and then contour plot it. And that's useful because now we can go into this plot with the experimentally observed relaxation time ratio, factor of 2 here, for instance. This is inspired by actual polystyrene PMMA blocker polymer values. And so we mark that contour value. Then we've done experiments on a neat polystyrene PMMA, and from that we can estimate that their relaxation times are in this range. So that gives us these possible ranges here on the vertical scale. This one is more tightly constrained because T1 average can be calculated from the observed relaxation time. So we have more knowledge here. And then we mark the overlap range and go down to the x-axis, read off the domain size and its range. That's still divided by square root of T1 average. We multiply that up. It's typically a factor near 1, so no big effect, but we can do it absolutely quantitatively and conclude, get a fairly tight constraint on the domain sizes. Each composition has its own plot, fortunately, and that's by doing it as a function of the slow relaxing layer thickness. They are quite similar, so they're easy to interpolate with. We don't need many of these to get meaningful values. And shown here are examples, again, from polystyrene PMMA, where we are fortunately in a quite steep region of the contour lines and get fairly narrow domain size ranges. So that's one way if you invest the time to find out about your intrinsic relaxation times. Now I want to move towards the simplest case. Maybe we have wood and we can't get the pure components, and we would still can obtain domain size informations by going into a limit effectively of infinitely long relaxation for the longer relaxation time. This is again a contour plot of the ratio of the observed relaxation times. Slightly different here because TB over TA, that goes to infinity. This one can be infinitely long, while the inverse is fully covered by the range from 0 to 1. Here, the 2 times are the same. We have no contrast and we learn nothing. Here we have a longer relaxation time of infinity, and that's why that ratio is 0. So it's the full range, and we can see that over this full range, a given value of, or measured value of the observed relaxation time ratio corresponds to a limited range of possible domain sizes. If this ratio is 2, you will never be able to observe that in a domain with less than 34 nanometers times square root of T1 average of that size. So each value of this observed relaxation time ratio is associated with a different lower limit. So we can make that into this graph. Here, the observed relaxation time ratio, here, the lower limit, and the lines for different compositions, fortunately quite tightly bundled here, so it doesn't matter if our composition knowledge is a little bit fuzzy. Here's a realistic example from an industrial polymer system that we've studied. We have a ratio of observed relaxation times of 1.45. We go to the x-axis, go up to the curve for our composition. It's about 50-50. Go over to read off lower domain size limit of 24 nanometers times square root of T1 average, which in this case was 1.6 seconds, so a little bit of an up correction. And we can conclude that in this material, we have 30 nanometer domain sizes, and we did not have to make any complicated assumptions about intrinsic relaxation times. I want to try to quickly show you how we validate this in commercial block copolymers, where the domain size can be tuned by changing the molecular weight from 80 to 2000 kilograms per mole. These are immiscible polymers that are covalently linked. So they want to separate, but they can't. They make these microphase separation layers, alternating layers, whose thickness increases with molecular weight. And so these systems, through head core analysis that I will briefly review, we get the domain sizes and then can fit the inversion recovery. And what you can already see here is that these realistic fits of simultaneous relaxation and spin diffusion are better than a simple exponential function as shown by the dashed curve. So this really supports that that's the right approach. Here, a little bit more about these block copolymer systems and NMR domain size analysis. It's a plot, double logarithmic plot of long period versus molecular weight. These are historical data. And in our work now, we're pushing beyond the long period of 100 nanometers and also doing it through inversion recovery. We use head core with proton spin diffusion to calibrate the domain sizes and the spin diffusion so that in the inversion recovery, only the intrinsic relaxation is somewhat unknown. We take slices at the aromatic proton chemical shift of polystyrene, shown in red here. And then we've developed a procedure of matching the full spectrum obtained, for instance, from projection of the 2D spectrum at the polystyrene peak position. And then we measure the fraction of the PMMA exchange peak here relative to that full spectrum peak. And this allows us, it's basically a B over A with an additional B full over BA normalization that allows us to, with very small error margins, get experimental data, even if we are far at the longest times from reaching the asymptote of equilibration and determine the domain sizes here. And I want to point out that with good enough signal to noise and decent spectral resolution, you can push into the 400 nanometer range with proton spin diffusion. We've done this for four selected block copolymers. You see the data here. For one of them, we also measured the spinning frequency dependence and found the expected slowdown of the diffusion at faster spinning, which allows for some tuning of the diffusion here. And that head core analysis gave us the domain sizes that we then put into the inversion recovery analysis. And this page compares the domain sizes we get from head core and from these contour plot analyses in inversion recovery. And the agreement is quite good. So to conclude, I wanted to show you that this old experiment is actually worthwhile to do, specifically by measuring spectra at the zero crossings. And that gives us spectra of domains in the material. And then we just take the ratio of these zero crossing times to get a lower domain size limit. For instance, 10 nanometers in that example of the wood that I showed earlier. If you want more accurate domain size estimates rather than just the lower limit, try to find out one intrinsic relaxation time through the experimentally measured T1 average. That's enough to give you a constraint of the domain size average through our contour plot analysis. And generally, I found it interesting to see that proton spin diffusion and NMR properly analyzed can actually get us domain sizes up to the wavelength of light. And with cylinders in a matrix, this is even by about a factor of 2 larger. There were some challenges to overcome and some technical points, but you've seen those. I want to acknowledge my co-workers, Chechen and Juan, for their help in this project, and Dr. Sommer at Firmenich Corporation for support of this work. And here one last page, an outlook, an off-spin of these inversion recovery experiments. We added a mixing time, realizing that this can be viewed as selective excitation. And we get exchange peaks during that mixing time. We can use the same, all the concepts from the analysis that I've shown you and the simulation techniques, and combine what these contour plots from inversion recovery with an independent measurement of the exchange peak experiment from the stair experiment that's shown in blue here. And they are nearly orthogonal and give us narrow ranges of domain sizes, and allow us to use this kind of approach to measure domain sizes up to about 300 nanometers without input of intrinsic T1 at ambient conditions. Thank you. So, let's see.
Okay.
I'm trying to find the question and answers.
Okay, well, they're right right here, Klaus. Okay, so
Okay.
Jeffrey, do you want to handle the question and answers? Jeffrey, you were going to do this for us today.
So, sorry, I had disconnected my microphone, and you can't see me either. I don't know how to handle this. I've never done this before, so I don't think this is a very good idea. But I can, of course, read them off. That's easy. So I see a question. I go from the bottom to the top. Is that no? From the top to the bottom. So Dominik Kubicki has a question. What other experimental techniques can you use to corroborate the domain sizes you determine from NMR? For example, in the case of the mixture of PS and PMMR.
Yes, so that was at the end of near the end of my talk. We use headcore NMR, so traditional, looking for cross peaks of headcore NMR here, transfer of magnetization from aromatic protons to the PMMA protons and carbons, and quantitatively analyzing this. And so this is a exactly justified question, and that's why we chose these model systems where that can be done, where both experiments are possible, and where we can validate that we indeed have agreement between an established technique and our new inversion recovery analysis.
But can you do the same, Kubicki asks, with a non-NMR technique, some refraction or whatever?
Yes, so in this historical work, you see small angle X-ray scattering, transmission electron microscopy, and there are lots of data for this system in the literature that weren't linked to NMR at all. So we could, so here for these large domains, it would be microscopy. It's probably outside the range of small angle scattering. But we feel that what we're mainly testing is that the spin diffusion part of our analysis is correct, and whether what we are obtaining from headcore here is just an effective model that gives the right spin diffusion, or actually the right model doesn't make a big difference for our test, because our test, we just want to make sure that we get the spin diffusion part right, and then we have the relaxation added. So it just needs to be consistent.
Then there is a question from Brad Schmelka. Can you comment on whether the T1 resolution is sufficient to obtain information on the interfacial regions of the two copolymers or distributions of domain size?
So the interfacial region, that would be difficult here. We are measuring one relaxation of all the PMMA segments, whether they are in the bulk or at the interface of the PMMA layer. The last one, that's actually the stair experiment is sensitive enough that we can see distributions of domain sizes. And there we're using the inversion recovery and the spin diffusion, and that is the only one that I'm aware of where if there's a broad distribution, we see a broadening of, we see a first fast increase of the exchange peak due to the small domains, and then a very dragged, but it never gets that high, and very dragged out decrease from the large domain. So in this experiment, we can see that.
There's a question from Walt Masevsky. In liquids, it is possible to partially tame spin diffusion with selective saturation. Would it be useful to selectively saturate individual proton chemical shifts during the inversion recovery to improve the estimate of intrinsic relaxation time?
So typically at our fairly slow spinning speed, I don't think we have the resolution to selectively saturate specific protons, but it's an interesting idea, something we should think about more. And one can do this at somewhat faster spinning where at least partial resolution is obtained. So thanks for that suggestion.
And Joe Freitas asks, he first comments, thanks for the great talk. And the question, what can you say about methods based on inversion recovery / spin diffusion without CP? For instance, in carbon-13 enriched materials with low proton content.
We have done this experiment on carbon-13 enriched materials, all on carbon. That's how we actually, in particular this one. So we do, so then you can simply do, because you have carbon spin diffusion in those systems, you can simply do analogous experiments with carbon rather than protons. The diffusion coefficient is about a factor of 1,000 slower, but you have seconds rather than milliseconds there. So then I would simply do it all on the carbon side, and we have done that, and it is useful. The domain sizes are smaller because of the slower diffusion, but it does work, and it does give interesting insights. We studied carbon-13 enriched nanothreads, and we saw perfect threads in a matrix of defective threads, and we were able to quantitatively estimate using this inversion recovery, and also then with external mixing, estimate the length of the perfect thread segments. So with carbon-13 enriched materials, very much on the carbon side, interesting to explore.
And then there is a question from an anonymous attendee. Does the proton spin diffusion coefficient also depend on the magnetic field of the spectrometer?
In the slow MAS limit that we're working in, where we have wide line proton spectra, it should not make a difference. When you get above 15 kilohertz, then you start to resolve the proton lines, and then the way in which you slow down the spin diffusion due to different overlap, that would change with field strength. At our conditions, it should be very minor. But when you spin faster, then you can again use it to tune. You do slow down the spin diffusion, so you lose the ability to see very large domains, but you get better resolution of the small ones.
So those are the last questions from the Q&A file. Thank you.
So Klaus, can you say, I mean, you've assumed a quote domain size. So you're assuming the domain is spherical, or?
No, these are known to be layered. That comes from both theory and electron microscopy observations. They're layered when they're 50-50. So these two systems are layered. When they're annealed, and we do anneal them and carefully, so NMR can show that at the conditions often used in literature, they actually decompose. These are at the edge of where you would expect layered structures, and these might actually have some cylindrical character. So based on the ratio of the polystyrene PMMA, it may change the morphology, but we are mostly near in a layered structure regime.
Mm-hmm. And so D then refers to the breadth, the thickness of the layer? The
thickness of the layer, exactly.
Yeah. Okay.
But we're also pushing into cylindrical structures and spherical domains.
Yeah.
Requires larger lattices, but we have ways of handling that.
Okay. Okay. All
I
right.
just ask one quick question. So I'm just curious, like, how would you expect things to change if I were to do it at, DMP conditions, like low temperatures and adding a radical?
I don't know enough about what happens to the relaxation times and how the radicals would affect it. Various groups, including Lyndon Emsley's group, of course, have done very nice work using the spin diffusion from this environment of the radicals to probe similarly large particle sizes in particular. But one part of my talk went into the head core, and I said, if you have good sensitivity, you can push into the 400 nanometers. DNP, of course, is a great way of getting good sensitivity. So that aspect alone, I think, is interesting. And that does not, head core works at all at temperatures. So I think that's an interesting combination that could be explored.
Thank you.
Okay, so probably we should move on to the next lecture from Galia. And Galia was an undergraduate at Colby College, where she studied chemistry and mathematics. And she was a PhD student here at MIT, and then did a postdoc with Tom Muir at Princeton University in a very biochemical lab where she learned some really interesting biochemistry. And following that postdoc, she assumed a professorship at UC San Diego, and she's been working on chromatin since the time she was with Tom Muir. And today, her talk is entitled NMR Adventures in the Chromatin Landscape. So Galia.
Okay, just a second here to share my screen. Can you all see my screen?
Yes, yes, okay.
And you can hear me well. Okay,
Yeah.
Thank you very much for the introduction, and thank you very much for the opportunity to be here today and to tell you about our work in using magic angle spinning NMR spectroscopy and some solution NMR to study complex biological systems. And we are switching gears, but for those of you who are interested in polymers, hopefully you'll recognize that the proteins and the systems that I have in my presentation, they actually have some properties of polymers, and we use some theory from polymer chemistry to actually understand their behavior. So my group, just a second here. So in my group, we are really interested in, well, 3 different topics. One of them is chromatin structural biology. Then we also study the phase transitions of neurodegenerative proteins. And we are also developing targeted DNP approaches for structural biology in cells. And these topics might sound like very different topics, but they're actually quite connected. And recently we have also started looking at the connection between neurodegenerative disease-related proteins and gene regulation. So today, however, I will focus mostly on our chromatin work. And since we have quite a diverse audience, I should start by telling you a little bit about chromatin. So chromatin is a way to package and organize the genome in the cell. So each one of our cells has about 2 meters of DNA. And this DNA needs to fit into a tiny, tiny nuclear space. So the nuclear diameter in a mammalian cell is about 10 micrometers. And the way this is done is the DNA is wrapped around very positively charged proteins called histones, and it forms the structure. Let me find my laser pointer. And it forms this structure here called the nucleosome. And this already provides a level of compaction, an order of compaction of 7. But then these nucleosomes can organize themselves into higher order structures forming chromatin. And then this chromatin is organized in the nucleus into different domains and of course chromosomes. So on a molecular level, the main building block of chromatin is the nucleosome. And this here is a beautiful crystal structure of the nucleosome. And you can see 147 base pairs wrapped around the protein core. The proteins inside, the histone proteins, there's 4 different histone proteins, H2A, H2B, H3, and H4. And there's 2 copies of each. So there's a protein octamer inside. And you can see that the nucleosome core is very nicely organized in rigid alpha helical structures. However, the histone tails, the C and the N-terminal tails, are disordered and they reach out of the nucleosome and they're dynamic, heavily post-translationally modified. And these post-translational modifications really regulate how the nucleosome interacts with proteins that are involved in gene regulation. And of course, I would like to point out here, this is a crystal structure, so we know very well what's happening in the core, but these tails here that are shown, that are drawn, they're actually imaginary. We don't know their position from the crystal structure because you cannot see them by X-ray crystallography or cryo-EM. And then on the very global nuclear level, chromatin is organized into 2 major domains. So if you stain the nucleus and take an electron micrograph, you will see these darkly stained regions, which are called heterochromatin, and that's where all the silence genes are located. And then you will see these lightly stained regions called euchromatin, where the active genes are located. And so in my lab, we are really interested in understanding the connection between the molecular and the global nuclear level of chromatin organization. And in particular, how do molecular events on the nucleosome level drive the separation into silence and active regions? And on the other hand, how does the environment in these regions affect events on the molecular level? And so for today, we'll focus on heterochromatins